Hello everyone, welcome back to our discussion on mutations. Today we are going to see the discuss about the molecular basis of induced mutations. So induced mutations are those mutations which are induced by external agents like chemicals, radiations, etc. And those molecules which causes mutations are collectively referred to as mutagens. So these mutagens induce mutations by at least three different mechanisms. Either they can replace a base, or they can alter a base so that they specifically mispairs with another base, or they damage the bases so that they no longer pair with any other bases in the normal condition. So the first of the category is a base analog, which substitutes the normal base. So this base analogs are chemical compound which are similar to nitrogen bases, and they incorporate into the DNA in the place of normal bases. Such bases are called as uh, base analogs. So an example of a base analog is shown here. So here what happened is that this is a pairing which is an AT pairing in a normal the DNA strand. So here it was A, and here you have this thymine here, and they are connected by two hydrogen bonds. So what happened is this adenine is now replaced by A star. A star is nothing but an analog or a base analog of adenine. So this base analog of adenine can pair with thymine, but in the next round of replication, you can see this is strand 1 and this is strand 2. They are replicating. So strand 2 does not have any problem. It incorporates into the complementary strand and adenine, which you can see here. But this particular first strand, this particular base analog, which was incorporated earlier, which is A star, an analog of adenine, what it does is that instead of pairing with thymine, it pairs with cytosine. So now what happens in this strand is from here, the base pairing in this position is going to change. So naturally, the base pairing in that particular position was an AT pairing, and because of the incorporation of the base analog from now on in the descendant generations, in the all the generation that will arise from this particular DNA strand, now the base pairing will be changed in this position to GC, and that is a mutation. Okay, such analogs or such changes induced by base analogs are called as, or that causes mutation. So here what happens is the original base analog exists only in one strand, and that is here the second one. Okay, and but it can cause a nucleotide pair substitution that is replicated in all copies of the descendant from the original strand. That is what we said. From now on, AT is replaced with GC in all descendant strands. So an example of that base analog is 2-aminopurine, which is an analog of adenine. And this analog of adenine, as we said earlier, this analog it can normally it pairs with thymine, but it also mispairs with cytosine when it gets protonated. And this AP is incorporated in the DNA pairing. What happens is pairing with thymine, it can generate AT to GC pairing, which is transitions by mispairing with cytosine and in subsequent replication. So this is what is shown here. So here you can see. So this is 2-aminopurine. It can pair with thymine in normal circumstances, but because these analogs are unstable, they easily gets protonated. As you see here, it gets protonated, and on protonation, it is pairing with cytosine. And because of that, an AT pairing is now replaced by a GC pairing. Because in the next round, the cytosine will surely pair with a guanine, and that is how a mutation occurs by this particular base analogs. And next, the agents are called as alkylating agents, and alkylating agents, they cause something which is called as a specific mispairing. So alkylating agents, they modify, they do not incorporate or they do not substitute nitrogen bases. They are modifiers. They modify the existing normal nitrogen base. So alkylating agents, they add alkyl groups. They will be adding alkyl groups to this to the normal DNA bases. The example of alkylating agents are ethyl methane sulfonate (AEMS) and nitrosoguanidine, which is NG. So they operate in this particular pathway, and what they do is they just modify the normal bases by adding alkyl groups. Okay, they can add alkyl groups in any of the four bases, and an example is that when oxygen is added to a position in guanine, it creates something which is called as O6 alkyl guanine, and this addition leads to direct mispairing with thymine. So that is also shown here. So here you can see this what happens here. So this is a normal guanine, and because of this alkylating agent, when this acts, you can see the incorporation of alkyl groups to the sixth position of oxygen. At sixth position of oxygen, this alkyl groups are, so here at sixth position of oxygen, this is free. Now here, because of this ethyl methane sulfonate, the alkyl groups are added, and when alkyl groups are added, it is pairing with thymine. So now the opposite happens here. That is here, a normal pairing was GC pairing. After this alkylation, now this will be changed to an A T pairing. So this is also a mutation, because from the next generation, the position, that particular position becomes mutated. So here also the same thing. This is another example where thymine is being alkylated. When thymine is alkylated, it is now pairing with guanine. So that is also a mutation due to this particular agent. So these are the first two. So the first one was a substitution where a chemical group or a chemical uh, non-natural analog of a nitrogen base is being substituted. Second one was modifiers, where they are adding alkyl groups or alkylating agents which add alkyl groups to the normal bases, and they cause mispairing. Now the third one is intercalating agents. So these intercalating agents, what they do is they do not modify the nitrogen base, or they do not substitute the nitrogen bases. They act in a special way. So these intercalating agents, they intercalate into the DNA, or they are called as DNA modifiers. So these groups include the examples like prophyllin, acridine orange, and a class of chemicals called as ICR compounds. They are intercalating compounds, and these agents are planar molecules. They mimic base pairs and are able to slip themselves and intercalate into the stocked nitrogen bases at the core of the DNA double helix. So, in this intercalated position, such an agent can cause insertion or a deletion of a single nucleotide pair. So they actually do not cause a mutation like what base analogs did or what alkylating agents did. What they do is that they intercalate into the DNA, and after intercalating into the DNA, what it does is either they will add a normal nucleotide into this DNA strand that also causes mutation. We said the insertion of a base, which is an indel or a frame shift mutation, or they can delete a particular base, and that is also an indel or a frame shift mutation. So that is how intercalating agents act. Next is tautomerism or tautomers. So these tautomers also are cause of mutation. So nitrogen bases are usually not static, and their hydrogen atoms, they move from one position to another in the normal nitrogen bases. And this chemical fluctuation, so the structure changes, or there will be structural alterations in the normal base because of this shifting in position of hydrogen, and such forms are called as alternate forms are called as tautomers, and the shifts are called as tautomeric shifts. So the amino form of normal uh, nitrogen bases is the amino form of that is adenine and cytosine are in their amino form, whereas thymine and guanine are present in their keto form. That is a normal state or a normal stable forms of this nitrogen bases. So this stable forms at times, not always, they undergo this tautomeric shift to less stable. Amino will be converted to its imino form, and the keto forms will be converted to enol forms. That means our adenine and cytosine, which is usually found in the amino form, will be converted to this imino. Okay, and the keto forms, guanine and thymine, will be converted to their enol forms. And these tautomers always all alter the normal base pairing rule. That is, adenine pairs with cytosine and guanine pairs with thymine. So this shift occurs during replication and this results in mutation. So this is the shift that is caused by. So this is not by any other agents like alkylating agents or our base analog, etc. So this happens naturally under such some conditions where tautomeric shifts can happen, and tautomeric shifts also results in mutation. So next is base damage. So base damage is usually caused by physical agents like radiation, and we are going to discuss about two main kinds of types of radiation. One is non-ionizing radiations like the ultraviolet rays, and ionizing radiations like the X-rays. So a large number of mutagens damage one or more bases, and so no specific base pairing is possible. So base pairing does not occur; it is damaging the DNA. So the result is a replication block, and the DNA synthesis will not proceed after apart from the base, and they cannot specify its complementary partner by hydrogen bonding. So first, we are going to talk about a non-ionizing radiation, which is the ultraviolet rays. So the difference between non-ionizing and ionizing radiation is that non-ionizing, both radiations, or both this ionizing and non-ionizing radiation, what they do is they cause, or they halt or they stop a replication process, because from the point where this particular radiations has acted, replications will not, or replication fork will not move forward, because there is a break in that particular point, or there is a halt in that particular point. So it can either be a break, that means when an ionizing radiation is hitting onto a DNA, it will break the DNA double helix. But when a non-ionizing radiation like the ultraviolet rays hits the DNA, that also result in a replication block, but it is not by breaking the DNA strand. It is by producing or altering something, or altering the basis. So we'll see how that is altered. So this causes or damage to the nucleotide bases in most of the organisms. So this penetrate to a number of distinct and alterate by the formation of something which is called as photoproduct, because it is from light, ultraviolet, right? And these mutations which is formed, they are called as photoproduct. So what ultraviolet rays usually does is that, so if this is a DNA strand, and for say there is a region where I'm having two or three repeated thymines. So there are regions where you have two thymines like this in the DNA. Yes, so here also I take two thymines in both the strands. So now what happens is, in an non-ionizing radiations like UV radiation is hitting this particular DNA strand. So what this UV radiation does is, so naturally what happens is this A will be pairing with T in the opposite strand. So here also you can see this T will be pairing with A in the complementary strand. So when this UV light hits onto the DNA, what happens is that this thymine or thymine which is close by, instead of pairing with adenine, it changes its pairing with or it loses its hydrogen bonding with adenine in the complementary strand, and the adjacent thymine, they pair up and they form thymine dimers. And this is called as photoproduct. As a result, there is no hydrogen bonding. And when a replication fork is replicating the DNA, it will halt from this position. Replication will not move forward. Okay, this is how UV radiations are causing the halt in replication or mutation. So this results in the formation of products like cyclobutane pyrimidine dimer and 6,4 photoproduct. So these are usually called as, or a general term for both this is called as thymine dimers, and usually this occurs between two thymines were close by. So from that particular point further, replication will not happen, and that is how it is blocking or it is causing a mutation in the DNA. So the last one is the ionizing radiations. So as I said, ionizing radiation, this act by two ways. So whatever the way be, this causes a break in the DNA. The first thing is that this particular ionizing radiation, they result in the formation of reactive oxygen species. Reactive oxygen species, they damage or they produce double-stranded breaks in the DNA, and also these radiation can directly cause the breaking of the backbone of the DNA. So the aqueous nature of the biological system generate effects by ionizing radiations in water, and it causes the formation of reactive oxygen species. They form different radix and also degradation product which damage the DNA. Ionizing radiation also cause direct damage to the DNA rather than through reactive oxygen species, and that is they cause double stranded breaks in the DNA, uh, and and leading to the formation of apurinic and apyrimidine site, and this causes strand breaks. So strand breaks are responsible for most of the lethal effects of ionizing radiations. So this is how the radiations or the physical agents. So we discussed only two physical agents. An example of non-ionizing radiation, which is UV radiation, and an example of ionizing radiation, which is normally the X-rays. So this is all about the molecular consequences of induced mutations. Now, so with this, we are coming to the end of the particular session, which is mutations. Now, so thanks for hearing.